Growth in bacteria isn't just about getting bigger, it's about making more cells. Unlike human cells, bacterial growth refers to an increase in both cell size and cell number, allowing a single organism to rapidly turn into a large population in a very short time. Once conditions are favorable, bacteria don't waste time. They divide by binary fission, a simple but highly efficient process. The key concept here is generation time, the time required for a bacterial population to double in number. For example, E. coli can double every 20 to 30 minutes, explaining how infections can escalate so quickly. Binary fission begins with elongation. The bacterial cell increases in length, preparing itself for division. During this stage, metabolic activity is high, and the cell readies all the machinery needed to copy its genetic material. Next comes DNA duplication. Using DNA polymerase, the bacterial chromosome is replicated precisely, forming two identical copies. This step is critical. Every daughter cell must receive a complete and accurate copy of the genome to survive. After DNA replication, separation begins. A new cell wall, called the septum, forms in the middle of the cell. The membrane and cell wall grow inward, eventually dividing the bacterium into two genetically identical daughter cells, each capable of repeating the cycle. Outside the human body, many bacteria can be grown in vitro on artificial media. These media provide essential nutrients such as sugars, amino acids, and salts, allowing bacteria to grow and multiply in the laboratory. This principle is fundamental to diagnosis, culture, and antibiotic sensitivity testing. Nevertheless, not all bacteria are metabolically independent. Obligate intracellular bacteria, such as chlamydia, depend on the host for energy. These organisms steal ATP directly from host cells, making them impossible to grow on artificial media and more difficult to treat. To further understand how bacterial populations behave over time, we use the bacterial growth curve, which measures the number of viable cells against time. This curve is divided into 4 distinct phases. The first phase is the lag phase. Here, bacteria are adjusting to their new environment. Cells are metabolically active and increasing in size, but not yet dividing. Enzymes and cellular components are being synthesized in preparation for growth. This is followed by the log phase, also known as the exponential phase. During this period, bacteria divide rapidly and at a constant rate. This is the phase where bacteria are most sensitive to antibiotics, especially those targeting cell wall synthesis, such as penicillins. As nutrients begin to run low, the bacterial world changes mood. In the stationary phase, growth no longer means expansion. Some bacteria divide, while others die. Waste products accumulate, the environment becomes toxic, and survival turns into a delicate balance. The total number of cells appears stable, but only because every new cell formed is matched by another cell lost. This is the phase where bacteria switch from growth mode to survival mode. Eventually, the balance is lost. Nutrients are completely exhausted, and toxic metabolites overwhelm the environment. In the decline phase, bacterial death exceeds bacterial multiplication. Cells lose their ability to maintain vital functions, membranes break down, and the population steadily collapses. What remains on the culture plate is a reminder that growth cannot continue forever without resources. This growth pattern is not just a laboratory observation, it explains what happens during infection. During the lag phase, bacteria adapt quietly inside the host, and symptoms are absent. As they enter the log and stationary phases, bacterial numbers rise sharply, toxins are produced, and clinical symptoms appear. Fever, cough, and fatigue reflect peak bacterial activity. When immunity or treatment gains the upper hand, the decline phase begins, leading to recovery and healing. Understanding the growth curve helps explain disease progression and timing of symptoms. Let's next discuss in detail the various requirements for optimal bacterial growth. For bacteria to grow at all, they must meet specific chemical requirements. Carbon is the backbone of all cellular structures. Nitrogen is essential for proteins and nucleic acids. Sulfur and phosphorus support enzyme function and energy metabolism. In addition, bacteria require ions such as potassium, magnesium, and iron, as well as growth factors like vitamins and amino acids. Without this chemical toolkit, growth simply cannot occur. Bacteria differ in how they obtain carbon. Autotrophs are self-sufficient, using carbon dioxide as their carbon source along with inorganic materials. Heterotrophs, on the other hand, depend on organic compounds, deriving carbon from preformed molecules such as carbohydrates, lipids, and proteins. Most medically important bacteria are heterotrophs, relying on the host environment for nutrients. Some bacteria are particularly demanding. These fastidious organisms cannot grow on simple media and require special growth factors such as blood, vitamins, or amino acids. Without enriched media like blood agar or chocolate agar, they simply refuse to grow. This nutritional dependence becomes a powerful diagnostic clue in the microbiology laboratory. Oxygen availability is another critical factor shaping bacterial survival. Aerobes require oxygen for energy production and thrive in its presence. Anaerobes find oxygen toxic and survive only in its absence. Facultative bacteria are flexible, able to grow with or without oxygen depending on environmental conditions. This diversity explains why different bacteria colonize different body sites. Surviving oxygen requires protection. Aerobic bacteria must defend themselves against toxic oxygen radicals. Enzymes such as catalase break down hydrogen peroxide, while superoxide dismutase neutralizes superoxide radicals. Without these enzyme defenses, oxygen becomes lethal. These enzymes are not only vital for survival, they are also essential diagnostic tools in the laboratory. Physical conditions matter just as much as chemistry. Most human pathogens prefer a temperature of 37 degrees Celsius and a slightly alkaline pH between 7.2 and 7.6. These preferences reflect adaptation to the human body. Temperature is one of the most critical factors controlling bacterial growth because it directly affects enzyme activity and membrane stability. Most human pathogens are mesophiles, thriving best at approximately 37 degrees Celsius, the normal body temperature, where their metabolic processes operate at peak efficiency. When temperatures drop below the optimum, enzymatic reactions slow, growth becomes sluggish, and bacteria may enter a dormant state. At higher temperatures, proteins denature, membranes destabilize, and cells may die. However, not all bacteria live by human standards. Psychrophiles are adapted to cold environments and grow best at low temperatures. A key example is Listeria monocytogenes, which can grow at 4 degrees Celsius and is responsible for foodborne infections associated with refrigerated dairy products and processed meats. On the other hand, thermophiles prefer high temperatures and use heat-stable enzymes to survive conditions that would kill most organisms. They are rarely human pathogens but are important environmentally and industrially. An example is Geobacillus styrothermophilus, which survives high temperatures and is used as a biological indicator to test autoclave efficiency. Its heat resistance underscores the need for rigorous validation of sterilization protocols. pH is a silent but powerful regulator of bacterial growth. As we have mentioned, most bacteria feel most comfortable in a neutral environment, with an optimal pH around 7.4, very similar to human blood and tissues. At this pH, enzymes work efficiently, membranes remain stable, and metabolism runs smoothly. Move too far into acidic or alkaline extremes, and growth slows or stops. But not all bacteria follow the rules. Vibrios are the rebels of the bacterial world. They thrive in alkaline environments, preferring a pH between 8.2 and 8.9. This explains why they flourish in seawater and why alkaline media are used to selectively grow them in the laboratory. Understanding pH preferences doesn't just help bacteria grow. It helps microbiologists choose the right media, predict habitats, and make accurate diagnoses. In the end, everything comes together. Generation time explains how fast bacteria multiply. Binary fission explains how they reproduce. The growth curve explains population behavior. And clinical correlations explain disease progression and recovery. With these concepts mastered, bacterial growth is no longer confusing. It becomes predictable, logical, and clinically meaningful.